This Week in Virology, the podcast about viruses, the kind that make you sick. You're listening to TWiV, the weekly discussion show all about viruses. I'm Vincent Racaniello, and this is episode 179. Today is April 13th, 2012. It's Friday the 13th. I hope nothing bad happens here. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How are you, Alan?
Doing well.
Nice
Things
day
are
today.
good.
Nice day, isn't it?
Absolutely glorious. It's bicycling weather, having a great time.
There's not a cloud in the sky here over the Hudson River. So everything been fairly reasonable with you?
Yeah, things are okay.
I like your post over at alandove.com about colony collapse.
Thank you. Yeah,
Nice.
I wasn't going to blog about it, but the news stories were just all over the place. I said, okay, fine.
Colony collapse is one of those things that tickles the news, doesn't it?
It really does. And this, I guess, just, it fit with a lot of people's preconceived notions, and so it became big news. And so I got a copy of the paper and looked into it.
Cool. Also joining us today from North Central Florida, Rich Condit.
Hi, fellas.
How are you
How
there?
you doing?
Rich?
I'm good.
You know, Rich, many years ago, I mentioned your name to Peter Palazzi, and I called you Richard Condit. And he said, no, it's Rich.
Oh, is that right?
Yeah. Is that what you like to be called?
Yeah, it's always been that. You know, I can tolerate just about anything but Dick.
You don't like Dick? I know someone who's called Richard Dick.
Weird.
I mean, that's a tough name. I don't know
It's
what
like
his parents were thinking.
major.
What
Yes.
is his parents thinking, you know?
Yeah, right.
Also joining us today, not too far from you, Rich, in the scheme of the world, from Georgia, the state of Georgia here in the United States of America, Gertrude Radu. Welcome to TWiV.
It's a pleasure to be here. Thank you for inviting me.
Now, you are a postdoc working at the CDC, is that right?
That is correct.
But you're not an employee of the CDC.
I am not. I'm contracted out through UGA, through a lab at UGA.
UGA, University of Georgia.
Yes,
And that would be Ralph Tripp's lab, right?
that is.
How
yes.
does that work? Well, let's start from the beginning. Where'd you get your PhD?
I got my PhD at Georgia State University in Margot Brinton's lab. And then when I was about to finish, I told her that I wanted to work at the CDC. So she actually had some connections. She had a friend, Larry Anderson, who at the time was the chief of the Division of Viral Diseases at the CDC. And he had told her that he was looking for a postdoc. So she put me in touch with him, and he then put me in touch with one of the labs that he was collaborating with in the it's RVIT, Respiratory Viruses and Immunology team. And the PI there is Leah Haynes, and they work on RSV. So he put me in touch with her, and then I went on a couple of interviews, and then basically I ended up working in that lab, and I'm still there. But one of our other collaborators is Ralph Tripp at UGA, and my work is actually funded through his grant. So I'm a UGA employee, but I do all my work at the CDC, and I live in Atlanta.
So who do you actually report to, Tripp or somebody at CDC?
Leah Haynes at the CDC.
CDC.
She's my PI, yes. But we do have meetings and teleconferences with both Ralph Tripp and Larry Anderson.
Oh, so you do talk to him now and then?
Yes.
Is there such a thing as a postdoc at CDC, or do you have to do it this way, through an institution somehow?
Typically, that is how it's done. Yeah, you come in as a contractor, and then at some point you kind of, in some instances, you transition over into other positions.
So does that work? Do you like it?
Yeah, sure. Love it.
So you're working on respiratory syncytial virus.
Yes.
And you're going to be able to publish in all of this someday? Typical?
Yes.
Yeah.
Yes, absolutely. And I should probably say at this point that everything I say here is my opinion only. So
And not that of Anyone
CDC
else?
or UGA or Georgia State or anything else, yes.
Anyone in the world, in fact.
Right.
That's correct.
But other people are free to share the same opinion.
Exactly.
Yeah, we have a disclaimer on the website. It's our opinion only and not that of our employers.
That's right.
I'm not sure.
My opinion is that of my employer.
Yours is, yes. Where are you from originally, Trudy?
I was actually born in Romania. My dad is Romanian. My mom is German, Transylvanian. Transylvania is a region in Romania that has a large number of Germans that have been residing there for about 800 years or so. So she's German. And for that reason, we always spoke German at home. And then when I was 9, we moved to Germany. When I was 12, we moved to Australia. And that's where we all learned English. And then in 1992, we moved to the U.S. So I've been here for almost 20 years.
1992, and that was before you went to the PhD program, right?
Yes, I had 1 year left in high school.
Yeah, cool. When you said Transylvania is, I thought you were going to say Transylvania is where there are a lot of vampires.
I was thinking the same thing, but I wasn't going to say it.
Why not? Why not? Because you're more mature?
It could be. Could be. That's a good suggestion.
Did you ever see a vampire, Trudy?
All the time.
Are you one?
But we don't talk about that.
So you yourself are a vampire then, right?
Half. My mom is German. My dad is Romanian.
Right.
Wow.
You should have gone into hematology.
Yes. Well, why do you think why do you think I'm here today when we're talking about mosquitoes feeding on blood?
Right.
Yeah, there you go.
So this brings up an interesting question. With with vampires drinking everybody's blood, they must have had all kinds of antibodies and viruses and all sorts.
Somebody should sequence the vampirome.
Yeah, that would be quite
Good
revealing.
idea. Let's get right to it.
It's like sequencing bat guano. You get all the insect genomes of the bugs that they eat. It's the same thing with a vampire. Anyway, welcome and glad to have you. I know that you've been listening to TWiV for a long time.
Yes.
And we've corresponded on the social media. So I thought, why not bring you in?
Thank you.
There's something really important here that
What's
we're
that?
leaving out. Okay.
Wait a minute. Wait a minute. Let me think. All right, go ahead, Rich. I'm sorry. I can't think of what
It's 80 degrees in Florida.
Oh.
And blue skies, puffy clouds.
What's it?
Cool.
What's
That's
it in
about
Georgia?
26 degrees Celsius. Okay.
69 degrees Fahrenheit, 21 degrees Celsius, and it's sunny.
Ah, it's a lot cooler in Georgia, isn't it?
Yes, it's actually been pretty cool the last couple of days. Very,
Farther.
very strange.
Farther north. What about you, Alan?
We've got. Oh gosh, I forgot to bring up the weather page. I'm shirking my TWiV duties.
But you have an iPhone right in front of you, right?
It is 63 Fahrenheit, 17 Celsius, and it is clear blue skies. And I did mention that it's wonderful bicycling
Yes,
weather.
right.
So I picked my daughter up from school by bike.
63 in April in Western Massachusetts?
Yes,
Is that,
Wow.
Is that warm?
it is.
It does seem warm, yeah.
Here it is
Yeah, well,
exact.
it actually hit 80 a couple of weeks ago when we had that warm spell.
Here it's exactly 17 also, 17 degrees. Quite nice. All right, so let's get into our papers here. We have 2 papers dealing with insects. And I learned this week that there's no such thing as insectology, right, Alan?
Well, there's a journal dedicated to it, apparently, but I took an entomology class in college, and insectology never came up.
It's pretty funny.
There's the Bulletin of Insectology, and the term's used nowhere else.
Before we talk about insectology, we need to make 2 quick notes. One, they're both passings, unfortunately. One is Renato Dubacco, who died not too long ago. He was 93. 93, is that right?
Yeah.
Amazing. And, of course, Renato was the developer of the plaque assay for animal viruses, and also got a Nobel Prize in 1975 along with David Baltimore and Howard Temin. Now, I think Dubaccos cool because he was, at the beginning of the 50s, he was doing cell culture. It was a brand new technique, and he recognized how important it was. And he took it and he developed the plaque assay.
Yep.
And then Howard Temin, this was at Caltech, decided to do a postdoc with him. And there he took these cell cultures and he infected them with Rouse sarcoma virus. So he took chicken fibroblasts, put Rouse in it, and they formed foci. It was the first time that anyone had worked with this virus outside of chickens ever since Rouse had isolated it back in 1911. And that's where Temin got the idea that this RNA tumor virus was permanently modifying the cell. And he went on to his own lab and found reverse transcriptase. And then meanwhile, Dubacco said, I'm going to infect these cells with DNA tumor viruses. And he got foci, and he found that the DNA was integrated, and so T antigen was made. So this guy was at the nexus of so many things. It's amazing.
Yeah. And he's the D in DMEM.
He is.
That's right. Dubacco's modified
Eagles medium.
Yeah. And he probably doesn't make a penny off of it. Because in
he
those
was
days,
he was the director of the Imperial Cancer Research Fund when I was there.
Ah, really?
And so his Nobel Prize was announced when I was there, and we had a reception for him downstairs in the lounge. Drank some champagne. It was a fine event.
Did you ever talk to him?
Sure.
What was he like? He had an accent, right?
Yeah, he had an accent.
Was he patrician?
Nice guy. I didn't talk to him enough to get a real strong impression.
Huh.
He's a nice guy.
Yeah, so apparently he was doing experiments up to a couple of years ago.
Great.
So, that's cool. Anyway, that's a big loss. The other one was someone sent to me by Lynn Enquist. He said you should mention this, and this is William Jarrett, a veterinary pathologist who apparently discovered in 1964 the retrovirus that causes leukemia and lymphoma in cats. Has anyone heard of William Jarrett?
I have to say that this is a new one to me.
Yeah.
This was a new one to me, too.
I'm very sorry.
Yeah.
He was working in Glasgow, or in the UK, and he was the most distinguished veterinary pathologist of his generation. So, there you go. He's dead at 83. Feline leukemia virus. And so, 2 virologists. We age just like anyone else.
Indeed.
All right, let's do some papers here. We have 2, and the first deals with dengue virus. The title is Dengue Virus Infection of the Aedes aegypti Salivary Gland and Chemosensory Apparatus Induces Genes that Modulate Infection and Blood Feeding Behavior. This was, I think this was sent in by a couple of readers. It's by Sim Ramirez and Dimopoulos from Johns Hopkins. And as everyone knows who listens to TWiV, dengue is the most important mosquito-borne viral disease that infects humans.
Even if you just started listening last week.
Last week. Did we talk about it last week?
Dengue comes up all the time.
Yeah.
We did do it last week, didn't we?
Mm-hmm.
Yep.
Oh, that's right. The T-sharp. Of course. How could I forget?
Yeah, while we're on that topic,
Yeah.
I wanted to bring attention to the last sentence in that first paragraph in the introduction, where they say that vector control remains the best method for preventing transmission of dengue.
Yep.
And this is actually in contrast to what Tyler Sharp said, isn't it?
It is. He said that there's no genuine scientific evidence that vector control does anything.
Yeah,
Yes.
that's right. He did say that, yeah. But he did say that tequila was very effective.
Right. This may have to do with there being formal scientific proof that vector control, some sort of controlled experiment
Yeah.
that shows definitively that vector control really controls the disease. And apparently, that's the way I read it. Apparently, that evidence is lacking.
Yeah.
Well, but the other thing to remember is what are we comparing it to? I mean, what are the methods we have to stop a dengue epidemic besides vector control?
Right.
It clearly works for malaria.
Yeah, most effective.
So it's most effective because it's pretty much the only thing you can do.
But for malaria, it works great. You give screens to people's homes, and you really reduce the incidence of malaria in, say, Africa. So, it should work, but I don't know.
Right. We know from other mosquito-borne diseases that vector control is effective. So, by extension, it's probably safe to say that that's a good method for dengue.
Yeah. Anyway, this study focuses on the vector, the mosquito, in particular Aedes aegypti, and the primary vector. And as everyone knows, the mosquito brings the virus from person to person. That's mainly how the virus is transmitted. And an important part of the mosquito for doing that is the salivary gland. So when a mosquito takes a blood meal, I saw a great talk in Dublin about this. Someone in Manhattan, Kansas, who works on mosquitoes and viruses infecting them. And he showed great slides of taking blood meals. The virus goes into the gut, replicates, and then eventually spreads to the salivary gland. And of course, there the virus replicates. And then when the mosquito takes its next blood meal, the virus goes out into the host. And as Dixon de Pommier says, the mosquito salivary gland is a pharmacopoeia. It has all kinds of drugs. It has anticoagulants, anti-inflammatories, vasodilators. I love this sentence. Vasodilators which facilitate probing and blood meal acquisition.
Yum.
Yeah, that sounds good. And of course, it has immune factors so that whatever it eats, blood or nectar, it doesn't infect the mosquito, right? And then they also say that there are factors that impair the immune response of the host, which is interesting.
It blows my mind that this little critter, the dengue virus, is adapted to replicate and spread in two completely different hosts.
Mm-hmm. Yes.
I mean,
yes.
Other insect-borne viruses as well, but this is a good example.
Yeah.
I mean, because he's extraordinarily well adapted to both the mosquito and to humans. It's amazing.
I think you've said that before.
Have I?
Yeah.
It bears repeating.
But you can repeat it. It's
senile.
fine. You're absolutely right. All of this is the product of selection processes.
Yep.
This is amazing. You can select for anything with viruses because there's so many and so diverse, right? So the point of this paper is to look at the salivary gland, to basically do microarrays, to look at RNA transcripts in the salivary glands. First, to see if they're any different from the rest of the mosquito, and then to see what the virus does to it. So they're basically taking parts of the mosquito, dissecting them out, making RNA, and applying it to microarrays to answer that question. And the first thing they do is to look just at the salivary gland and see if it's unique, any unique transcripts compared to the rest of the body of the mosquito. And they find, in fact, a number of genes that are transcribed specific to the salivary gland. Let's see. 2,255
right,
salivary
2,255 are enriched in the salivary gland.
Yeah,
Right.
15% enriched in the carcass relative to the gland. That's the carcass, and 2,255. And then they break these down into what they think are interesting. So there are a couple of groups. One is enzymes, 24 enzyme or transcripts involved in digestive functions, which makes sense, right? Because that's what the salivary gland in part does. It puts out saliva that helps you digest things. Alpha amylases, glycosidases, proteases of various kinds. And so these, of course, could help digest the blood meal. These, again, are specific for the salivary gland. There are a lot of genes involved in immunity. A variety of antimicrobial peptides, proteases, innate immune genes like toll-like pathway genes. And they speculate that these are probably involved in the defense of the mosquito because, again, you drink blood, you can get all kinds of things when you do that. And then there are others that were interesting to me because I hadn't thought about these. Blood meal acquisition genes. There are some genes in the mosquito whose gene products make you itch and burn after they bite you. And then the mosquito counteracts that. So you won't slap it, I guess.
Right.
So you itch, and the mosquito is putting things in you to stop you from itching, like anti-inflammatory enzymes. Incredible. Anticoagulants also, so that you don't clot up and make it impossible for the mosquito to keep on drinking.
Right.
And these are all found in there. Proteins that bind biogenic amines like serotonin, histamines. It's just amazing. And then there are a bunch involved in chemosensory functions. Smell and taste. So they have odorant binding proteins, which are carriers for odorant molecules involved, I guess, in the mosquito smelling its host. And
because you got to find your host.
Taste it. So
And this is a little weird because you got the odorant binding proteins in the salivary gland, right? And they go on about that a little bit because that's not ordinarily where you smell, but they must have a function there.
Right.
So these are like, aren't they found in the lymph of the insects?
Yeah. They think that it helps carry odorants through the various structures to the nerve endings that smell. What do you call those things again? Come on.
I don't know.
The
I don't know.
nerve
olfactory?
Thank you. The olfactory receptors, yeah.
Oh, okay.
So
I knew that.
They're found to be transcriptionally downregulated in salivary glands of some mosquitoes after blood feeding, which suggests that their function has something to do with the feeding, I guess.
Right. Yeah. So it's some kind of satiety signal for the mosquito, probably.
Right.
Satiety means you've had enough to eat, Alice?
Right. You already ate. Don't go looking for another blood meal. Although after mosquitoes feed, they usually can't go looking for another blood meal because they're so bloated they can't really fly. So they just kind of sit there and digest.
I'm thinking of a Far Side cartoon where a mosquito hits an artery.
Right.
Oh, yeah, that's great. That's a good one.
So do you think evolution has gone wrong there? Somebody can just come along and squash them?
Oh, when they're digesting?
Yes.
Well, enough of them survive.
Yeah, there's so many of them, I suppose.
That's that's the basis of evolution. Is it doesn't matter if some go.
Right.
And there isn't really a good way for them to solve this problem otherwise. They have to somehow get enough nutrients to be able to lay a clutch of eggs. So that's going to have to come from a certain load of blood, and then they're going to have to digest it.
So here's a question for all of you. What gender mosquito are they studying?
Female.
Females.
So all this is female specific, but males do have salivary glands, right?
They eat fruit juice.
They eat fruit juice. I wonder if you compared them, if you'd find any differences. Right? Because the females have evolved to seek a host and eat blood. You would imagine that something is different.
Yeah.
From what I understand, this has been barely studied in mosquitoes.
I'd be curious to see what a mosquito Iacocca protocol looks like.
Yeah, they talk about how they, followed other protocols. I mean,
I know I read
they have
that.
an ethics statement.
Oh, ridiculous. Probably it's a 100-page protocol to do a mosquito experiment.
A nightmare.
This at this New York Academy symposium on H5N1 some time ago, Arturo Casadeva was complaining about having to fill out a 100-page protocol to do experiments with mice, but he could go to Home Depot and buy a mousetrap and kill as many as he wanted at home.
And then he can't publish it.
At this Dublin meeting, I hate to keep diverting, but at this Dublin meeting, this guy talking about mosquitoes, he was doing this one experiment where they had to put electrodes into the mosquito. So they had to rip out all of its legs. So it was just a body. I really I felt bad about this.
I had friends that did that in grammar school.
He had a picture of this one mosquito, with a probe going in its proboscis, I guess, and it had no legs. It's just, I know that this is just a mosquito, but I just felt bad.
I feel absolutely no compunction about anything that's done to a mosquito.
You think I'm misplacing my...
No, I'm with you. I'm with you on this, Vincent.
Yeah,
considering what mosquitoes do to people, I don't know. I
don't say we shouldn't do it. I just felt bad. It pushed the feeling bad trigger somewhere in me. I'm really.
I can see. Yeah, I can see your uneasiness with that. I do understand that.
I've killed mice by the thousands. It doesn't bother me at all.
But you didn't cut their legs off.
No, I didn't keep them alive with their limbs off. Right. I would anesthetize them, and a lot of people have problems even doing that. But
yeah, we culturally, we have a lot higher tolerance for things like that being done to more distant species from us. Right. So if you're doing it to an arthropod, then it's not considered as bad. We don't generally throw chickens in the pot whole and boil them, but that's perfectly acceptable when you're cooking up some crabs.
I remember my parents used to put lobsters in boiling water. I hated that. It really bothered me.
On a tangent here, that whole topic.
We don't go.
Wait, we're already on a tangent.
Now you're going to go on a tangent?
Sorry.
On a second tangent, just real quick. That whole issue, you guys have discussed this before, with smallpox. Do we consider the eradication of smallpox as something to mourn? Right. So I teach a freshman biology class at Georgia State, too, in my spare time. And last night, I actually had a student come up to me and ask me that question. It's like, do we look at it the same way as, like, would it be okay if we eradicated mosquitoes? What would that do to the population? Well, we wouldn't have malaria and all that. But, all these other animals would die. But what about smallpox? How has that affected the population besides the fact that we have more longevity now?
From an ecological point of view, it has a lot of impact. Yes, we have more longevity, but it also creates a vacuum where other pox viruses are entering. So that's interesting. But it also, just in terms of eradicating a species, when we talk about finally nuking smallpox, that comes up periodically, whether to get rid of the last of it. Invariably, the question comes up of we are deliberately extinguishing a species or an organism. How do we feel about that?
Right.
I don't have a problem with it.
Yeah, I've kind of swung around between both points on that debate, and these days I kind of feel that we shouldn't get rid of it.
You have smallpox.
Smallpox.
Oh, you can eradicate it though. That's fine.
Oh,
right?
oh yeah, get it out of people. That's fine.
Okay.
I'm fine with it not being in people, but I'm talking about destroying the last stocks.
Sure. Yeah, I agree.
Completely eliminating it, and it's just that's a totally irreversible decision, and and that's something that's I don't think we should do. I think we ought to hang on to it.
I don't think it's worth it because you can always remake the virus. If someone wanted to use it, they can remake it from the sequence.
But you can't remake all the ones that you didn't sequence.
Right.
All the strains you didn't sequence.
I agree.
There's a tremendous library of genetic diversity in those stocks. It's hundreds of vials.
Right.
And we haven't, if you've gone and you've sequenced all of that, and you really have gotten all the possible information out of it, and you've looked at the possible epigenetics of it and all that, then maybe. But I think there's more information in there that we could get out, and that's what I have a problem with.
I'm just saying from the point of view of anti-bioterror people, they say keeping it around is dangerous, but I don't think that's a valid argument because you could make it. Right?
I don't find the biodefense arguments very compelling anyway.
Yeah.
I'm immunized.
Yeah. When you went to CDC, you got immunized?
No, as a kid.
As a kid, yeah, I was too. You're not that old. Come on.
Romania. They
Romania.
stopped later there, yeah.
I see.
I'm immunized.
Yeah, I was immunized as a kid. And,
I've been immunized about a dozen times.
Yeah, you have been, and you should be. All right, back to dengue.
Back to dengue and mosquitoes.
Then they look at what the virus does. They infect mosquitoes with a blood meal. And they look at they do a microarray in the site of salivary gland, and they find 38 transcripts that go either up or down. And these have a variety of functions. I don't want to go into all of them.
Actually, it's 147.
147? Dengue altered the abundance of 38 transcripts with functions related to metabolic processes. 147 total. 38 with respect to metabolic processes, stress response, transport, cytoskeletal proteins. Also some immune function proteins, which you would expect that the mosquito is raising immune response to virus infection. And then there are a couple of proteins, cathepsins and cystatins, which are involved in regulating apoptosis. Which
have opposing functions.
Yeah, so that makes sort of sense. And then the ones that are really interesting are the odorant-binding protein transcripts. 2 of them, 10 and 22, they go up with dengue infection. And that's cool because maybe the virus is actually upregulating proteins involved in smell and seeking a host, which would be really cool. Another example, now when people say, do you know any virus that alters host behavior? Maybe we can add dengue and mosquitoes to that list. Although not yet, probably.
Not yet, yeah.
All right, so they take a bunch of these, a handful, and decide to knock them down by RNAi in the mosquito and look at the effect on dengue yield. And here for Rich Condit, here is the method. You're going to love it, Rich.
Yeah.
They orally infect with dengue through a blood meal, and then they inject 2 micrograms of double-stranded RNA per mosquito. They inject it into the salivary gland.
I'm visualizing
This is a really small thing, Rich.
I'll say.
And they have to inject it. They must do it under a dissecting microscope, I guess.
Yeah.
The mosquitoes have to be anesthetized, right?
Yeah, my guess is they knock them out with CO2 or something like
Can you
that.
imagine trying to hit the mosquito if it's moving around? Be like one of those mole. What do you call those mole things?
Whack-a-mole.
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 Thank you very much.
Well, the blood meal thing, they to infect them, they basically infect cells in culture with dengue and make a lysate and then mix it with this artificial blood meal. It's human blood and human serum that they feed them through a membrane. So they spike this blood meal with dengue and infect the mosquitoes that way.
Yeah. And so that the silencing is done at 7 days post blood meal. DPBM. That's the acronym.
Yeah.
So I have a question about that.
Yeah.
I was wondering, since the gene silencing is a transient effect, do we know how long it lasts?
I don't think they looked, right?
No.
They didn't address that. The reason I'm asking is because I wonder if prolonged silencing would eventually lead to, not to starvation, but to reduced breeding because the females need the blood for egg production.
Could be.
Mm-hmm. Yeah, it's good. They actually did not look at the target, as far as I know, right? They didn't actually confirm silencing.
They confirmed silencing.
They got a supplementary figure.
Did they have
here.
a Western blot or something?
Yeah, they did. No, they did confirm it, but they didn't look how long. I think they just looked
didn't
at
do a
one
time course.
Yeah, they just looked at one time.
As we've already pointed out, these are not simple experiments.
No.
I'm actually impressed by people who work on mosquitoes. After hearing some of this, it's really, cells are good for me. I don't know, this mosquito business would be too hard. All right, so some of these had effects on dengue titers. So silencing of cystatin and a protein with ankyrin repeats, which is involved probably in immune responses, increased viral titers. So you figure you knock down an immune-related gene, it's going to increase titers, right? That makes sense. Silencing of cathepsin reduced the viral titers. So some of them went up, some of them went down. The cathepsin is involved in apoptosis, I believe, right?
Yeah, well, the cathepsin is a protease, while the cystatins are protease inhibitors that regulate apoptosis.
So cystatins negatively regulate apoptosis. So if you take them away, you should get more apoptosis. And this made dengue titers go up, and they say maybe that's because it more easily spreads from cell to cell or something like that, right?
So inhibition of apoptosis by the cathepsin silencing would preserve the cells and then inhibit dengue virus replication through RNA interference.
Yeah, that's what they think. What other ones? Okay, so those are some
and these are not massive radical changes for most of these in viral titers. I mean, you look at the scatter on these plots, and there's
quite a bit of scatter.
Yeah, there's quite a bit of scatter, and then they average it out. But the statistics seem to more or less add up.
I think that's what you're stuck with when you do
exactly. You're
mosquito
dissecting out all these salivary glands, and
they have to do many mosquitoes per condition. It's
right, and they're going to vary in their amount of silencing too.
Yeah,
so they saw silencing.
Go ahead. Sorry.
Yeah, so they saw gene silencing. Some of them are 26%, some of them are 90% silenced. So there's variation from bug to bug.
They do between 60 and 80 mosquitoes per condition.
Right,
it's a lot. Then they silence these odorant binding proteins, and this is an uninfected mosquito because they want to know what this is doing, and they have this wonderful assay. That's called a behavioral feeding assay. So they inject the double-stranded RNA, and then 4 days later they do this feeding assay. They were offered a mouse. I love it. They were offered
an anesthetized mouse.
I just have this vision of a silver platter with a mouse.
Yeah, the choice of words is really funny.
Offered a mouse. And then they measure probing propensity, which is the percentage of mosquitoes that probed within a fixed time period, which is probing meaning biting the mouse. Probing initiation time, the time between when they offer the mouse and the mosquito bites it, and then probing time from initial insertion of the proboscis to the engorgement of blood.
which
And
sounds really
it sounds like they're doing most of this manually, right? Because they stick like 4 mosquitoes in. Because later on they talk about sticking more mosquitoes in and video tracking them because they can't do more than 4 otherwise. I can't imagine doing this.
Yeah, I think somebody sat there and with a stopwatch and timed everything to
Something
400.
like that.
Yeah.
Silencing of the 2 odorant binding protein genes resulted in a reduced probing propensity, which is, how many mosquitoes probe within a fixed time period.
Right.
It increased the probing initiation time, right? How long it takes for them to start biting. Probing time was also increased, but not statistically significant. So these seem to be involved in blood feeding, right? These 2 odorant binding protein genes. Yes, Rick.
I was just looking up here a little elaboration on the methods. I said they probably knocked them out with CO2. That's wrong. They cold anesthetized them.
Oh, yes.
To inject them, and then they dissected out salivary glands from these guys.
Yeah.
And did plaque assays on them.
Right.
I mean, really.
Vincent's favorite assay.
Rich, too.
Plaque assays, yes.
They're great.
Good.
I'm happy they didn't do PCR instead. Yeah, that's great.
Yeah.
A lot of people just hate plaque assays. They want to do PCR.
A mosquito's small enough, but salivary glands, well, I guess they've been doing
invasive.
that all through this, but still, it's a little hard to fathom.
Trudy, do you do plaque assays?
Yes.
You like
Good.
them? Do you like them?
Not my
Yes.
favorite.
You love plaque assays. You just don't know it yet.
Our plaques look a little different.
It's okay. It's okay as long as you have to learn to love them. I guess you will. You will. But would you prefer like a fluorescent assay or a PCR based assay?
Yeah, PCR probably.
See that?
How do you feel about mercaptoethanol?
Smells.
You don't like
You
it?
see?
No.
Wow.
You see, it takes a while. I love the smell of mercury. As a matter of fact, I like the
Smells
smell
like victory.
of autoclaved biological waste. All right?
That's awful.
I can't help it. Yeah. All right.
All right. So when they silence these odorant binding protein genes, they say, the silencing isn't specific to the salivary gland. It could be in the other parts of the mosquito as well. So they go on a series of experiments where they look at the antennae of the mosquitoes, which would also be expected to have these OBPs in them. And they find that the antennae are, in fact, infected with dengue virus, which seems to be surprising to them. The virus replicates in chemosensory organs. And these transcripts are enriched in those organs as well, the odorant binding protein transcripts. And they also looked at transcripts encoding a subunit of the odorant receptor, the actual receptor that binds molecules and transmits a signal to the nerves. And those are increased as well by infection. So the virus, not only in the salivary gland, is inducing also in the antenna genes that are involved in host seeking.
So there's a lot of circumstantial evidence here leading them to the idea that maybe the virus is influencing the behavior of these creatures.
That's right. So we haven't had a direct experiment. And the last thing they do is actually to test that, whether dengue infection heightens chemosensory abilities. So they infect mosquitoes with dengue or not, and then they look at their blood feeding behavior. Present them with a mouse.
And interestingly, in this experiment with the dengue-infected mosquitoes, they present them with a dead mouse rather than just an anesthetized mouse. And they have to keep the dead mouse on a little heating pad. Okay,
Yeah. They lose body heat
keep them
very
warm
quickly.
enough so that the mosquitoes are still interested.
So why do they use a dead mouse for this assay?
Well, it must have something to do with must be a biosafety thing having to do with the dengue. I don't know. I mean, I'm just guessing.
Wait, but in the figure legend, they say mosquitoes were offered an anesthetized mouse.
Because I thought I read in the
say.
methods. Go ahead. I'll check this out.
All right. Anyway, there's they find slightly shorter probing initiation and probing times in infected mosquitoes versus controls, but they say they weren't statistically significant, and there was no change in probing propensity. So this is the key experiment to see if the virus has an effect on these properties, and they don't really see very much of an effect.
It didn't really work.
Here you go. Behavioral assays involving dengue-infected mosquitoes were carried out in 14 days, blah, blah, blah. A maximum of 7 mosquitoes placed in a cage. A Swiss Webster mouth was euthanized via the administration of an overdose of ketamine and offered to the mosquitoes. To counteract the drop in body temperature, a heat pad was placed over the mouse for the duration of the assay. And here's what I was talking about before. Because of the larger number of mosquitoes per cage, video recordings were made during the assay to allow the experimenter to keep track of individual mosquitoes.
Those must be scintillating to watch.
Yeah, they're on YouTube. You can watch them.
Where's the popcorn?
Yeah. All right, so to wrap up this experiment, they say in the discussion that we didn't find a difference in the feeding behavior of infected versus uninfected, but it could be that in nature, we see small differences, and maybe in nature those would be amplified, and you would really see a big difference in the field because there are longer distances involved, and you can't really look at that in the laboratory. So they say small differences. Yes, go ahead.
Or it may just be that the virus simply takes advantage of a mechanism that already exists, and that being that mosquitoes feed on humans sometimes.
Yeah, I mean,
whether or not virus infection occurs.
Yeah, I mean, they don't. I think they don't have to induce any feeding alteration, right?
Right.
Yeah.
It's just a really interesting idea.
So in the end, we're not sure. We know that certain transcripts are induced by infection, which have the potential to alter feeding behavior, but whether or not that happens, we don't know.
Yeah, it could be entirely coincidental that the virus, in infecting these cells, it happens to induce these genes and a bunch of others, and yeah, these are involved with the odorant receptors, but maybe it's not really relevant.
Yeah. So what would be the experiment you could do to test this?
Well, first thing I'd do is do a live mouse instead of a dead mouse. I wonder if that makes any difference. Okay?
Yes.
It might be more appetizing to the mosquitoes. The other experiment that you could do is stick a human in the cage, but I don't think that is ethical.
That's not going to clear the committee, but no. I think Aedes aegypti has a preference for humans, and is that one feeding on birds or pigs? There's some other host that it likes to feed on, and it's not mice.
I don't know. We need Dixon.
Yeah, we do.
Not here. I mean, the mosquito of West Nile likes birds.
Right.
And takes humans when the birds aren't around.
Those are Culex species. Culex. But I don't know here. I mean, you could modify mosquitoes so that they don't induce these odorant binding proteins upon infection, but I don't know if you could release I don't know if you could do that because there would be a chance that they would get out and maybe there would be concerns about them. But you could somehow modify them so that or maybe even take away the odorant binding proteins and see the effect.
Yeah.
So there are other things you can do. I'm sure these guys are going to be doing this. Anyway, this got a lot of attention. There's a Times article about it as well because viruses changing behavior is appealing to people, right?
Mm-hmm.
Yeah.
All right. Anything else on this before we move on? Trudy, any notes that you want to share with us?
No, I like that title for that Times article. A virus may make mosquitoes even thirstier for human blood.
Yes.
Very sensationalist.
Yep, that's what the writers do. The science writers.
Exactly.
It sells papers.
They have to sell. Pretty soon, they're not going to sell any papers.
Well, right.
And then what are they going to do? They haven't figured it out.
Would you like fries with that?
What are they going to do? All right, next paper. Now, I tell you a little history here. I first met Rich Condit on a study section 20 years ago, probably. Right, Rich?
Early 90s, probably 90, 91. So, yeah, over 20 years ago.
So I first met him, and he's reviewing grants. He's got this raspy, deep voice. And then he gets this grant on a virus of a wasp. And he prefaces with, he says, "This is just the coolest story I have ever heard. Just stay with me here." And he goes on to tell this story of this wasp virus, and that's what we have today. So you really like this, don't you?
I really like this stuff.
I love it.
Yeah, this is neat.
So I'm going to let you tell us.
So I've been sort of casually following this ever since that first episode. I I was the pox guy on the study section, so they couldn't. There's not a lot of pox grants, so they couldn't really fill up my dance card all with pox. So I got all the weird viruses because I would do the homework. So this is a polydnavirus. And I don't know how to pronounce some of this stuff, but the official name is Bracovirus. I guess that's Bracovirus, not Bracovirus, right?
Yeah,
it's all made up. It's made up. What's the difference?
Bracovirus.
Right. Okay. Well, I don't want to offend anybody. So, I should start off by saying that this the paper that we're going to talk about here was published recently in Journal of Virology. The authors are Burke and Strand from the University of Georgia. And I should say that it's actually follows up on an article that I was not familiar with because I don't follow it closely enough, that was actually published in 2009 in Science magazine, where the authors are Bézier and Dresden, based in France, with some collaboration from some people in Switzerland. There's a lot of authors. So that was the lead author and this senior author. And there's a commentary that was published at about the same time. So before I get to the papers, let me give some background here. So the polydnaviruses, they're called polydna, that's poly-DNA viruses, because they package multiple different, and by multiple we're talking 30 to 50 circular DNA molecules in their capsid. These are viruses that appear in parasitoid wasps.
So, Rich, they're 30 to 50 per capsid, or one per capsid?
One per capsid. I think, well, there may be multiple in capsids. I'm not sure they even know that. But if you collect a whole bunch of virus and extract the DNA and look at it, you get out 30 to 50 different circles. I'm not sure that the packaging is sorted out. But there's enough DNA so that I doubt if you're packaging them all in a single capsid.
Do you need all 30 to start an infection?
Well, that's...
we probably don't know.
They don't really infect it,
Well, they get into cells and express genes, right?
Yeah, right. Okay, so let's back up here.
Yeah.
We got first of all, the biology of the wasps. These are parasitoid wasps. So these wasps lay their eggs
inside a live caterpillar.
Inside a live caterpillar. These are caterpillars of moths or Lepidoptera, moths or butterflies.
Yeah, so this is the story behind Alien.
Yeah, exactly. And so they lay their larvae in the caterpillar, and the caterpillar is effectively dead or paralyzed or something, but stays...
Paralyzed. It's alive, but paralyzed.
Yeah, it stays biologically active. And the larvae develop inside the caterpillar, and then actually come out of the caterpillar and pupate. They spin cocoons that are hanging all over the caterpillar. Okay, so they pupate outside the caterpillar and then fly off. So there's these wonderful pictures that we could use maybe in the episode of these caterpillars with these cocoons hanging all over them. It's really gross.
Yeah, so they devour the host from the inside while it's alive.
Yeah.
And then they hatch out and put their pupae on the outside. And
So the deal is that when these wasps are developing and making eggs, there is also a virus that appears in the oviducts of the female wasps. And you never see this virus anywhere else. Okay, and it is laid along with the eggs, and the virus then infects the caterpillar cells, and the genes that are, I'm being very careful with my vocabulary here, the genes that are encapsulated in the virus are expressed in the cells of the wasp that the virus in the cells of the caterpillar that the virus infects, and they have immunosuppressive effects on the caterpillar, because the caterpillar is going to mount an immune response to the larvae, and the virus then basically helps out the wasp by suppressing the immune response of the caterpillar to the larvae.
That's something we don't have any idea about mechanisms, right?
They can identify some of the immune modulators that are used here. That's and I don't know a lot about the mechanism, but I think there is something known.
So this is a symbiotic virus of a parasitoid that helps it infect its host.
Right.
And it's only vertically transmitted.
Yes.
Yes. Okay. So the deal here is that I'm avoiding the use of genome, really, in talking about the DNAs that are packaged. Because the strange thing is that these DNAs that are packaged encode what are essentially wasp genes with immunomodulatory properties. And a lot of them, in fact, are not even like, they're like got introns and all kinds of stuff. They look almost like they're fresh out of the genome in terms of their structure. And none of these genes encode any viral replicative genes, functions like, DNA polymerase, capsid proteins, RNA polymerases, all of the sort of nuts and bolts that you would expect for viral replication. And in fact, when the virus infects the caterpillar, there's no virus replication that goes on. It's just delivering these immunomodulatory genes, which, if I understand it correctly, actually integrate into the DNA of the cells that are infected in the caterpillar and express the immunomodulatory functions. So this is a gene delivery thing. Now, not only that, but these genes that are encapsulated and delivered are in all of the somatic cells and the germ cells of the wasps. Okay, so what happens is that during the development of the wasp, or during the life cycle of the wasp, these genes are just in all the somatic cells and the germ lines of both males and females. But it's only in the female at the time when the oviduct is making eggs, that these genes are amplified out of the wasp genome and packaged into these capsids, so that they can be delivered along with the eggs.
So there's some signal during egg development that turns on.
Must be some sort of
hormonal.
Yes, a hormonal signal during the egg development that turns on this whole process. But as I said, none of the genes that are encapsulated encode any structural proteins or anything else. So in the 2009 Science article, the Bezier and Dresden article that I mentioned, the investigators looked at genes that were expressed in the oviduct of the wasp using an EST tagging method. So they're looking at messenger RNAs in the wasps. And what they discovered was that there were a number of genes that were expressed in the oviduct that were related to a different virus called a nudibvirus, which is related to baculoviruses. And these are insect DNA viruses that are kind of standard, large nuclear DNA viruses that just do their virus thing. And they found several genes that looked like they were related to this, and they're saying, aha, maybe these nudibvirus genes that in fact are integrated also in the wasp genome are supplying the functions to encapsulate these immunomodulatory genes and do the virus part of this whole life cycle. So the article that I found that was published recently in JVI by Burke and Strand follows up on this, and they do first they set up a system. They're using a different parasitoid wasp than was used by the previous group. This is Micropletus demolator. Am I doing that right, Alan? You're
Sounds
the
good to me.
Here, okay.
Yeah.
Demolator?
Demolator
or
okay,
demolator,
fine,
I'm not sure which.
Fine. they're using that particular wasp, and they set the system up really nicely. The first thing they do is to do assays, a time course assay through the development from larvae, or actually from pupae through adulthood, looking at the amplification and excision of the DNAs that are going to be transferred in the oviduct of these wasps. So they stage the wasps, and they look at different times through the life cycle, and they find that the DNAs are the DNAs that are going to be delivered are amplified actually in the later stages of pupation. And this must be some sort of onion skin amplification, because they're amplified like orders of magnitude before they're actually excised during the late stages of pupation. And then during early adulthood, early and mid-adulthood, then they're actually excised and circularized. And that's at the same time as you're making a bunch of virus, and they show up in the oviduct. So that tells them when to look for expression of genes that might do all this stuff. And then at an appropriate time during the development of these creatures, they do deep sequencing and look, and they say, we're going to look and find whatever is there, either wasp or viral, and we're going to look for what's amplified. And what they find is that these wasps express, about 44 different genes that look like nudibvirus genes. Mm-hmm. And they include a couple of things that are involved in DNA replication, a heligase and an integrase. Notably absent from this are DNA polymerases and a lot of other things associated with DNA replication, and we'll get to that. 4 different RNA polymerase subunits that are like nudibvirus genes, transactivators involved in late gene expression, several nucleocapsid proteins, things involved in infectivity by feeding, a couple of other structural proteins, and other nudibvirus genes of unknown function. So apparently, at some different location in the wasp, there have been integrated ancestral, these must have been from a long time ago, because nudibviruses, if I understand it correctly, don't ordinarily integrate their DNA. But there must have become integrated into the wasp genome these nudibvirus genes that are specifically amplified at the time that you need to amplify and encapsulate these immunomodulatory genes, and they function to do just that. These genes are turned on and probably work together to transcribe other genes and make the capsid genes, and et cetera, and encapsulate the immunomodulatory genes, which are independently being amplified and excised for encapsulation.
And they say that this probably happened over 100 million years ago.
Yeah.
Because that's when the wasps diverged, right?
It's been around forever.
Wow.
So, as I said, what's missing from this whole thing is DNA replication genes, and they presume that, in fact, the DNA replication functions are supplied by the wasp, and they play around with trying to figure out which those might be, and they come up with I think it's polymerase gamma, a mitochondrial DNA polymerase. I'm not picking
Delta?
Up. Delta. Okay. So we got basically 2 different sort of origin, 2 different bits of what you might call genome, except one doesn't get, this is bizarre. We got an integrated pieces of an ancient virus that turn on genes to encapsulate genomic segments that by a different mechanism are amplified along the same time scale and excised, and then all encapsulated to be laid along with the eggs to do the immunomodulation with the in the caterpillar.
So this is really a hybrid, right? It's a hybrid of nudibvirus-like capsid proteins and Bracovirus gene fragments.
Well, yeah, but at this point,
Yeah,
what's a Bracovirus?
Yeah, exactly.
All right. A
Yeah, I think-
Bracovirus is basically amplified wasp immunomodulatory genes. They're obviously specialized for this because they have sequences on them, flanking them that are specific for their amplification and excision and packaging in these capsids. So they're specialized for this purpose. But is that a virus?
Yeah, they're just circles of DNA with genes on them, right?
Yeah, what I think happened here is there was this ancient nudibvirus infection that accidentally got integrated. And the byproduct of that was that suddenly these wasps gained the ability to transmit initially random bits of their DNA.
Right.
Because they had this viral machinery that was now encapsulating and just packaging up bits of DNA and sending it around. And at some point, some wasp, randomly gained the ability to.
Did we lose Alan?
He's gone.
Oh, I thought there was something wrong with my headset.
Randomly
No.
gain the ability
Yeah.
to to immunosuppress, but that had to happen quickly, otherwise the DNAs would have been lost. I think there has to be some selection to keep them there, right?
Yeah, I mean, this is it makes my head hurt to try and think about it.
So if on that question, is it a virus? Is hepatitis delta virus a virus?
Well, we actually have a different name for
Yeah, they
that,
call
right?
it viroid, right?
Right.
Yeah.
I mean, they have a VIR in the name. They're virus-like, satellites, viroids.
Right. This is just like totally different. So what this paper, to me, the new thing in this paper is, well, as I say, it's not entirely new. It's just better developed. Is the idea that these nudibvirus genes that are integrated elsewhere in the genome are what are responsible for assisting in amplifying, well, not really amplifying the DNAs, but somehow at least making the capsids, probably through a fairly elaborate process. The actual amplification of the DNA is probably done by the wasp.
Right.
So this is a symbiosis, right?
Yeah.
Both sides win, right?
To the nth degree.
Yeah. The wasp is benefited, and what does the virus get out of it? It goes to.
It gets to keep being undead.
I guess so. It can propagates in the wasp, right?
Yeah. It's
Yeah,
an
I don't really see this as being.
It's not. So the big question is it a virus?
Yeah, I don't see this as a virus anymore. It was at one time, clearly, but after it integrated and the wasp put it to this use, it's really kind of a wasp system
This
now.
is just gene transfer, really.
Exactly. It's wasp gene therapy.
It is.
Yeah. So it essentially gave the wasp a pilus.
I mean, the only reason it stays in the wasp genome is because it confers an advantage to the wasp.
Right.
But it's not. So if you want to say it's selfish DNA that it's perpetuating itself, then in that sense it benefits, I suppose. But
Yeah, but all DNA is selfish in that sense.
yeah,
I really want to call it a virus because I want to keep it in virology. Okay.
Otherwise, we can't talk about it here.
That's right. It's just too cool.
well, we keep
There's no question, but that it has a viral ancestry.
Right.
it has a capsid too. It's got capsids, right?
And it has a capsid.
So that's fine. I mean, we talk about prions in virology courses, and they have nothing to do with viruses, but they're cool. They infect, they replicate. So I don't think you have to worry about it.
So how many more pieces of biological machinery scattered throughout nature have actually been the byproducts of viral infections?
Yeah,
Good question.
probably a lot.
We're looking at a system that we happen to be able to see thanks to this work, and thanks to the fact that enough of the viral genes remain in more or less intact. But if it were doing something else that didn't require the capsids, a lot of this stuff would have evolved along with the genome in its own way.
Sure. Of course, there's the integrated retroviruses with placental formation, right? So we can see that. But there are many others, I'm sure. And if they happened a long time ago and there are no more particles, you wouldn't see it. But this kind of thing, I bet, goes on in many other systems. We just haven't looked. Yeah. All right. This, I suppose, was stumbled on because someone found the particles. But if you carried out a search through all insects, for example, or even plants and other species, you might find more of them.
Yeah.
Why not? There's never just one of anything, right? One thing in this 2009 science paper I wanted to point out, Rich, they did purify these particles, and they did mass spec, and then they lined up the sequence with the sequence from the wasp genome, which showed that the capsid proteins originated from the wasp.
Right.
So that was an important link between the two. Well, it's a great story, yeah. This is a huge sequencing effort, basically.
Yes.
That's what this is.
And also, from a technical point of view, keep in mind that these guys are raising all of these creatures in their laboratory and then dissecting out the oviducts from the wasps and from wasp larvae at different times during their development. So this is, it seems to me, a technical feat as well.
Go through a lot of caterpillars.
Yeah, a lot of people who like to do that combination of biology.
Did they mention their institutional approval protocol?
No.
Yeah, at what point don't you have to get it, right? You do need it for mosquitoes, right?
I had a difficult time understanding some of the methods. What is a hidden Markov model?
Ooh.
Oh, yeah, that goes way beyond me. Forget it.
I mean, I tried to look it up, and I found a dissertation.
Yeah.
Right. No, I have to, in particular, when it gets into some of the sequence analysis and that kind of stuff, I short out. I just have to trust them on that.
Well,
I certainly did.
It's related to a Markov model, right?
Which is hidden.
Yes.
Right.
A Markov process.
So I was talking to McFadden about this, and he was friends with a guy who does polydnavirus stuff in Canada named Don Stoltz. And he said he visited him in his lab one day, and he says, let me show you. I'm going to do an experiment for you. And he takes this big, long piece of clear Tygon tubing with a cotton plug towards the end, and he sticks it in a cage and up next to a wasp and sucks the wasp into the tubing and then goes over to a cage with a caterpillar and poof, blows the wasp out onto the caterpillar and says, look, I just did an experiment.
Well, Trudy, these guys are from Georgia. Do you know them? You probably don't. They're in entomology, right?
No, I got my undergrad at UGA, but that was a really long time ago.
I see. A Markov process, by the way, is a random process, a stochastic process. I love this explanation. A stochastic process that satisfies the Markov property.
Oh, that's good.
Loosely speaking, a process satisfies the Markov property if one can make predictions for the future of the process based solely on the present state, just as well as one could knowing the process's full history.
Oh, that
got it?
Yeah, sure, I got it. That clears it up completely.
And this page is full of formulae. Here's a good one here.
Fun.
You can apply it to gambling, birth, all sorts of things. Oh, it's cool. Hey, check it out. Search for Markov process. All right. Anything else on that before we move on?
No. And I hope if I blew any of that, I hope the Polydna guys out there. I hope there's some Polydna people listening.
Are there any Polydna people listening? If so, make sure Rich got it right. That's okay. All right, let's do a few email here. First one is from Ibrahim, who writes, I was watching the conference in Dublin, and I wanted to thank you for sharing my email with the people in the panel, since I saw how nice it was for me to reach the specialists with that ease through TWiV. As a footnote, actually, my name is Ibrahim, not Lawrence, but thanks again. All right, so I read Ibrahim's letter in Dublin, but I called him Lawrence. So I want to publicly apologize for calling you the wrong name. I know that's really crummy.
He's got a little smiley face there.
He does have a smiley, okay. But I wanted to apologize because I don't want to do that. I think Alan should read the next one from Josh.
Sure. Josh writes, Dear Dr. Racaniello, thank you for reading my email on the air on TWiV 178. It's always nice to be included on my favorite podcast, even if you guys did disagree with me. Josh was the one who wrote about the H5N1 story, and looks like we won.
Right.
I think that was that letter. Would you mind an outsider's perspective on the situation? And although TWiV is my favorite podcast, I am an outsider. I'm not an academic, and I don't know any of the people involved. I am apprehensive about the entire role of the NSABB and fear that the biological community will soon be subject to the same levels of restriction and classification that currently affects the physics community. I think that in almost every case, things that fall into the category of classified information are almost always done for reasons that have nothing to do with science and everything to do with bureaucratic nonsense. Restricting information about nuclear weapons, for instance, hasn't prevented many countries from acquiring them. Which disappointed me when you read my letter on TWiV, not because you disagreed with me, but because I wonder if you were seeing the larger picture. I agree that it should be about the science, but it's not. Once you remove the scientists, and there are a lot of non-scientists on the NSABB from the equation, it's about politics and the bureaucrats. So they're going to either restrict the information or they're not. If they do, we agree that this is a bad thing. If they don't, this is a good thing. In this instance, it's kind of a zero-sum game. A game that I believe TWiV played a large part in winning, because week after week you applied pressure to the scientists and other members of the NSABB by calling them out on their nonsense. It's one thing to be rebuked for your actions behind closed doors, and quite another to be publicly reminded that you are not following the science by your peers. You had an issue that you were passionate about, you skillfully applied public pressure, and your side won the day. That's not only how you play the game, it's how good policy wins out over bad policy. The next policy fight that is coming is that those people on the other side want to change the entire scientific publishing system in the US and Europe. They want to have a system where those people on the list get the papers and everyone else does not. Those people on the list will not be able to discuss it either. Anthony Fauci said as much on that NPR story. It's not only bad policy, it's bad science. TWiV can either be in the middle of this policy fight or it can be on the sidelines. I know what I would prefer. I know it's distasteful. I know you don't want these public dust-ups, but what choice do we have? If you don't engage, then there will not be any opposition to the plans, and that's an outcome I don't think our country can afford. Sincerely, Josh. P.S. On a personal note, TWiV and your online virology course inspired me to sign up for MIT's new online course offering, 6.002x Circuits and Electronics. It's week 5 so far, so good. The math is hurting me since I haven't had calculus in 20 years, but I'm mucking through it. Okay. Yeah, so what I was objecting to in the idea of winning this is I don't think there's a winner or a loser in this whole securitization of biology. I think we all lose by having this situation where things were, I think, handled kind of badly.
I agree that we have to have a stance and talk about it and express our opinions. That's certainly not what we meant by not acknowledging the win. And I understand his point completely that someone has to speak up, otherwise the other guys win, if you want to put it that way. So we will do that. We will express our opinions, and I understand what you're talking about here. I personally don't. This is my opinion, not that of TWiV or my employer. I personally think we don't need the NSABB. I think we should just publish what we do, because I don't think anything bad can ever come of it. But I think we're stuck with the NSABB, and we have to work with it. I don't see it going away.
If we are going to be forced to have a process of that sort, then at the very least it needs to be something that occurs at the grant review stage and not at the publication stage.
Yes, that would be better.
Because this idea of having a paper that was done in unclassified settings and then it gets all the way to publication and it's been handled in completely non-secure fashion all along the way, and then you suddenly say, oh, we're not going to publish it. It's just wrong on multiple levels, and it's a real waste of time and effort, and I think that was an error.
So I'm with you on all of this stuff. My only issue with what transpired is that there were times during the process when it became personalized, ad hominem attacks, as we've talked about, and times when motivations were attributed to people, I think, potentially unfairly. And I think that when we have this kind of discussion, it's important to do it in a civilized fashion and try to avoid that kind of thing. and keep focused on the issue rather than the people. And as long as we do that, I think it's fine. And I think we can play an important role in outing the debate. And I also think that as I think Alan said, or I forget whether Alan and Vincent said, we're stuck with something like the NSABB. But in fact, what was originally recommended in the Fink report was to capitalize on the process that was put in place during the recombinant DNA debate and basically piggyback the whole bioterror dual use thing on that process. Where there's a local institutional review, and if problems come up like that, in that process, there are higher levels that you can appeal to, going all the way up to the NIH, and some, basically, I think they proposed using the RAC, the Recombinant DNA Advisory Committee, to do some of this. And the NSABB is a sort of a peripheral organization that can develop policy and help, if necessary, adjudicate or advise on some of this stuff. And, those are reasonable proposals, most of which were never followed up on, if I understand it correctly. And so ultimately it all fell onto the NSABB rather than going through this process. And that's one of the reasons it came out so sloppy. So maybe this will precipitate going back and revisiting this and putting into place a process. And I think it's important to note that when we did this with the recombinant DNA thing, and we had a process in place, it turns out that very, very few that we executed the process, and very, very few issues actually went all the way up to the NIH level Recombinant DNA Advisory Committee, and science has been able to go forward, and there hadn't been a problem. And my guess is that with respect to dual use, the same thing will happen, only it's got to be done. I mean, if there's a process, it won't be as sloppy as it was this time.
Right. And Josh, I think the disagreement that we have is not so much on your main thesis, which is, we ought to have these discussions and speak openly about them. I would agree. I think it was just the use of the term win was really the crux of the problem. Because when you phrase it in terms of winning and losing, then all these personal attacks and such tend to rear their ugly heads, like Rich was talking about. And it's not a process that I think we should frame in those terms. I think we should say, look, we all want this science to get done and done right and done safely. Let's come up with a sane and rational way of handling that, and the current system is not it.
I quit watching TV about 10 years ago, and one of the reasons I quit is that anything that passes for some sort is put up as some sort of real debate or conversation on the TV is now the paradigm is you get two radically opposed individuals who stand up and trash each other.
That's right.
Yes.
Okay.
Right. Yeah.
And that's supposed to be debate. And I don't want to do that. Okay. We need to set a better example than that.
Yeah, I agree. As you probably know, Rich, that HHS, Health and Human Services, decided that there would be review of certain kinds of experiments before they take place now. So there's a list of about 10 pathogens and about 5 different kinds of experiments, and they say these have to be reviewed, which is good depending on who is going to review it.
Right, and the science has to be on the top of that list. It's got to be done with the science in mind.
Yeah.
Right, but that's a process that, if it's structured even halfway reasonably, as Rich points out, there should be this appeal process. If you don't like the initial decision, you should be able to follow it up and all that sort of thing. And you can have all of these debates in the open, by the way, when somebody's proposing an experiment, because there's nothing classified about proposing an experiment. You don't have any data.
Yes, absolutely.
So if you want to have that conversation, that's I think should be one of the stipulations of that, by the way, is that those discussions people should be able to discuss freely.
Yeah.
And if I understand it correctly, this is no different than the process that was put in place for the recombinant DNA stuff. I mean, they talk about I remember, it's still the case that you have to with a biosafety committee. This is where biosafety committees came from. Basically, was the recombinant DNA debate.
Right.
And they're there to monitor initially recombinant DNA stuff. And now it's almost a rubber stamp because we have so much experience with it that there's hardly any recombinant DNA experiment that you can think of that's an issue. But it was the same process. Things are reviewed at the proposal stage.
I think the committee is crucial here. For RAC, it was right. It had the right composition because it was mainly scientists. And I know that's not going to happen this time. All right, there are going to be other people on this committee, just like there are on the NSABB. So,
well, we'll see. And
you're right, but I'm not optimistic.
I'm not averse to there being members of the general public or other people involved in such a committee as long as there's transparency.
I have a list of 10 people I don't want on it. All right. Trudy,
well, we'll see.
Any thoughts, Trudy?
I think everything should be published, including negative data.
Yeah, all right.
And by negative data, I don't mean my Western didn't work. I mean, like, incorrect hypotheses.
Yes.
Yeah.
That's a whole other ball of wax.
Boy, if I could publish all the Westerns that didn't work.
All right. The next one is from Tanel, who writes, Dear TWiVoners, which is TWiV plus marathoner. I'm a grad student from Freiburg, Germany, originally from Estonia, though, who has been listening to TWiV as long as has been running. On Monday 16th, I will run the Boston Marathon, and no matter how it goes, I would like to use the opportunity to thank the TWiV team for the support throughout the training season. Research and graduate studies can often be frustrating, and running has been a way to keep a clear mind. Running 6 times a week and between 8 to 30 kilometers a day means quite a bit of time in running shoes. TWiV and TWiP has made those hours also entertaining and educative. I'm sure there are many researchers in running shoes and maybe even those who will join me on Monday in Boston. Good luck, fellow TWiV listeners and marathoners. I thought we should read that today because the marathon is on Monday.
Yes.
Right. Good
Go
luck
to
in the
Nell.
marathon.
Good luck.
Yeah, go to Nell. Trudy, you must know, is Tanel
No.
what gender is that?
Oh,
you don't know?
I thought you were going to ask me from what country. Not German. What gender?
That's right. She's from Estonia.
Here.
So I was figuring with your German background, you might know.
I actually don't know the gender.
Okay.
I don't know.
At any rate, write us again and tell us how you did and tell us your gender.
Yeah.
Let's do the next one. We'll make that the last because this is a good question. We are at Alan, I believe.
Yeah.
Okay. So this is Maximilian writes, Hello, TWiVers. I discovered your netcast today, and I'm hooked. Cool. I'm currently a pre-frosh at Duke University's Pratt School of Engineering with an intended major in biomedical engineering. I have 2 questions for you, which I've been thinking about for the past few weeks while I finalize my intended major or double major. First, is there a possibility of working on viruses or immunology if I were to focus on protein engineering? I ask because my first love in biology was virology immunology, and that love has always competed with my more prevalent interest in biomedical engineering, BME. Would it be more prudent to double major in chemistry, concentrating on biochemistry, or biology, molecular and cellular, if I want my double major to focus on my interests in virology and immunology? Thanks so much, Maximilian.
Yes.
Yes.
Well, I think that a thorough understanding of biochemistry is imperative when it comes to studying virology. That's just my experience, though.
Mm-hmm.
But I don't know if you need to major in that. If you just understand just enough chemistry, and then just enough organic chemistry and then biochemistry to understand what goes on in the cell and the virology and all that, then I think you'll be fine. But
Do you think he should? So it's a choice between chemistry or biology as a double major. I would say chemistry.
Yeah.
Yeah.
Because I agree with you. If you know chemistry, you can do anything.
Training in chemistry is never wasted.
Yeah.
Right. And in the biological sciences, in particular biochemistry, I think of biochemistry as the language that we speak. And if you don't know, the better you are at speaking the language, the better off you're going to be. With respect to this idea of double majors and that kind of stuff, I would say, major where you're primary interest is, and then take the appropriate courses and stuff that will enhance that major. And if a double major falls out of that, fine. But, nobody in the long run really cares whether you had one major or two majors or anything like that. I don't think. Every now and then you can use that, but it's that's really not a big deal.
Well, the thing about having two is that you can creatively combine two areas, and a lot of people do that now because they think of careers where they want to use two different areas,
But you can always learn the other stuff later.
Sure.
Yes.
I suppose there is some advantage in having it officially down on a paper somewhere. Okay?
I mean, and in terms of learning the other stuff later, I would say get the chemistry in college.
Yeah.
I agree.
Because it's going to be harder to pick up later. Biology, I think, will be easier to pick up later.
I mean, I was a bio major in college, right? Just bio, nothing else. And I can tell you that the chemistry I took helped me much more in my current career than biology ever did.
Yeah, I was a biology major, and I discovered in my junior year that I really almost wished I had majored in chemistry, and so I ended up minoring in it.
Yeah, I was a biology major, and I had this. You can see where my perspective is coming from. I had all of the chemistry credits I might have needed to get a double major, except for doing a project or something like that, and that just wasn't worth it to me. So I was basically a double major in everything but on paper.
Right.
But the chemistry, the other thing I would say is that I wish I had paid more attention to the chemistry when it was happening. I didn't realize how important it was.
Exactly.
And there was a lot of it that I didn't understand, and I still don't understand.
What did you major in, Trudy?
Biology.
Yeah.
But I had a much harder time with chemistry. Like I really had to spend a lot of time on that. But now,
chemistry's tough.
in retrospect, now that I teach biology, I realize how important that general chemistry and that organic chemistry is at the beginning. I tell them just enough to kind of build on that and explain the macromolecules in the cell, but that is so important.
Right.
Yeah.
So we didn't answer his first question here. Is there a possibility of working on viruses in immunology if I were to focus on protein engineering? I would say absolutely.
Yeah,
sure.
Because people, I think there's a lot of potential here now for engineering viruses to work for us in one fashion or another,
Sure.
and that's gonna mean engineering proteins to do certain things.
Viruses or viral proteins for sure.
Right.
And you're going to learn a lot of the concepts that you would use in virology or immunology. So
Yeah, and in terms of working on viruses or in immunology, you'd be talking about a graduate degree. And by the time you get to that level, I went to graduate school with people who had majored in chemistry and with people who'd majored in, I guess the furthest afield was, I know a human geneticist who in college majored in math and tuba. He went to Peabody, so he's he's a professional tubist and a human geneticist. But the math is what got him into that, and so it's all statistics. So you can potentially do anything if you get a good grounding in the hard sciences. And if you come out with a biomedical engineering degree and a chemistry minor or a second major in chemistry or a biology major, all that is going to, almost not exactly fall by the wayside. You'll be building on it, but you'll, once you get to the graduate level, you'll be pursuing studies in that and you'll manage no matter what.
Speaking of double majors, my son wants to double major in history and computer science so he can design historically accurate video games.
Okay.
and what places he's visited, they think that's a great idea. So I think that's cool.
I think he's going to be rich.
I think he'll probably change his mind by then, but that's the idea of a double major. Yeah. Hey, this guy is a pre-frosh, which means he's a high school student. So maybe this is our fourth high school listener.
Yeah.
That's cool. I like that. Okay, let us pick some science picks of the week. Let's start with Trudy.
Yeah, actually, would this be a good time for me to talk about sequencing science?
We could do that.
Okay, after.
We could do it when we say goodbye. Yeah, you could fill us in on that, sure.
Right.
Unless you want to make it your pick, you could do that too if you want.
Well, I have a pick too.
Okay.
Okay. So Yesterday, I received the MMWR. That's the Morbidity and Mortality Weekly Report that the CDC puts out. And I picked this one article. It's called Rabies Risk Assessment for Exposures to a Bat on a Commercial Airliner.
I saw this.
And I'm going to call it Bats on a Plane. Mm-hmm. Well, basically what happened last August, a bat flew from the rear of a commercial aircraft through the cabin several times before it was trapped in the lavatory, and this happened minutes after takeoff. And there were 50 passengers on board. The pilots were notified. The aircraft was returned to the airport. And then of the 50 passengers, only 15 reboarded the aircraft, and the other 35 made alternative arrangements. And of course, the main concern was rabies, because this posed the question of whether or not post-exposure prophylaxis is needed in this case. But it turned out that none of the people had any physical contact with the bat or exposure to its saliva. So
they didn't immunize anyone. That was the decision,
right? But the other funny thing about this is that this is in the Madison, Wisconsin airport, and there have been previous bat sightings at that airport, and we are all going there in July.
Cool.
Maybe we should get our rabies shot.
I'm bringing my camera for sure.
They did say that they went through the airport and that kind of stuff and found that there was no particular bat infestation or anything like that, and everything was okay.
Yeah.
The other thing I liked about this was, of course, one of the deals that they wanted to do was get the bat and find out if there was actually any risk of rabies, but the bat got away. He flew out through the boarding tunnel and through the airport and out one of the doors in the airport.
They say through automatic doors.
Right.
So this was a smart bat. It waited for the doors to open and flew.
Well, it's a frequent flyer.
This is just great. I bet they had a lot of fun writing this, right?
Yeah.
Yeah, we'll be in Wisconsin. Maybe we'll have a bat on our plane. That's cool. That's really neat. Thanks for that. Rich, what do you have?
What do I have? Yes. The Giant Magellan Telescope. I just learned about this the other day. This is a project for building what looks like an amazing telescope. It's going to be in Chile. They've just started construction of the site that's going to hold the telescope, and it's at something like 6,000 feet.
This site is amazing. Yes.
Yeah. Very, very dry, great weather, so it's good seeing conditions, or perfect seeing conditions, as they say. And the mirrors it's a mirrored telescope, and it's an array of 7 mirrors, and the mirrors themselves are really interesting because they're made of I can't even describe it, it's on the website, a funny kind of glass that's very light. It's sort of some sort of fibrous thing that winds up with a polished surface, and it's a segmented mirror that they can actually warp the mirror in place, so they can make adjustments to the mirrors according to the conditions or whatever. So it's an enormously sophisticated telescope that is supposed to have a resolving power that's 10 times greater than the Hubble. So that'll allow them to see stuff way out there and get a lot of new information. It came to my attention because of an article saying that NSF had basically offered them a $250,000 5-year deal for development, or at least to apply for this. It was a request for an application, and they decided not to apply. They've got $25 million donors in that kind of stuff, and they have a long letter in here about why they turned down the NSF's request. And basically, it's very interesting. This is a huge consortium with people from Australia and the U.S. And elsewhere, and they're basically saying I guess NSF has already put a fair amount of money into it, but with respect to this proposal, they're basically saying, we can come up with that kind of scratch ourselves. If you guys are really interested in making a serious investment in this, then we'll listen. So at any rate, there's an interesting site, and I look forward to this thing being constructed and see what it comes up with.
It's an interesting consortium of universities
and
it is. Institutions. Wow. That's great. Yeah. All right. Rich. No, not Rich. Alan. Sorry, what do you have for us?
Well, mine is also on an astronomical theme. It's a YouTube video. It's an animation called "The Rings of Earth," and the jumping off point for this was the hypothetical question: what if Earth had rings like Saturn? And so it starts out with some views. It's just space view of the Earth, and they've drawn in very very nicely drawn in what the rings would look like from space. But then the cool part starts a couple of minutes in. They start showing different views of major landmarks and major cities around the world, and what the view would look like from the ground of these rings stretching off into the sky from different latitudes and different sites. And it's really, really cool.
Nice.
Yeah, I really enjoyed this. It is very nice.
And would it make any difference to Earth? Do we know?
Might. I don't know if it would affect tides or anything like that.
Yeah, I was wondering the same thing.
Yeah, there'd have to be some gravitational effects. They don't really go into all of that sort of thing. I just, but the thing that you would see is these rings just extending up into the sky. And if assuming the rings were at the equator, if you were at the equator, you just have a line going all the way up into the sky all the time. And if you were further up, you'd be able, if you were at northern or southern latitudes, you'd actually see a sort of an angle-on view of the rings just permanently in the sky, which would probably completely have changed our perceptions of space.
It would also cast a shadow, which I assume would move.
Yes,
around.
they show the shadow cast on the ground as well, and how it would be cast in different places.
That's very cool.
Yeah, it's very cool.
My pick is something I used this week. This week in my virology course, we had a talk about antivirals, and I used this site because we talk about hepatitis C virus, the new drug pipeline. And this is a site that summarizes every drug and the phase of research that it's in: research, preclinical, phase 1, 2, or 3, and approval. And it's just amazing to see how many different companies and different drugs are being developed to inhibit this fairly important human virus.
I was blown away by this.
Yeah,
I wonder what the one for HIV looks like.
That's a good one. I bet there is one for HIV. So, Alan, why don't you type in HIV new drug
I'm just
pipeline.
looking for that.
I mean,
I've seen that. I've seen that.
So
in various
competitive.
places,
Can you imagine how competitive this is?
Sure.
Oh, it is. In fact, there's been discussion in the industry for the past couple of years about how this might end up hurting some of these companies that are in this business, and inevitably it will. There are going to be some winners and losers here.
Of course.
I'm not finding any really good graphics for the HIV pipeline.
So there are two, of course, two new drugs recently approved, which we've talked about. On TWiV, two protease inhibitors, telaprevir and boceprevir, and then a bunch of others. Also, RNA polymerase inhibitors, more protease inhibitors, some siRNA inhibitors as well, non-nucleoside, nucleoside inhibitors. It's an amazing array of targets. It's really cool.
Yeah, some of these are going to come to grief, and some of them are probably going to succeed.
Yeah. Anyway, check that out. That's pretty neat. It's a nice graphic. We have two listener picks. One from our friend Ricardo in Portugal. It's called Evolution, the Natural History of Animal Skeletons Stripped Down. It's an article at Brain Pickings. What a flamingo, capybara, and a guinea pig have to do with the beginnings of recorded time. That's a pretty cool article.
Cool.
Thank you for that. Ricardo and Peter writes, hi, TWiVers. You'll probably get lots of suggestions for this, but I hope I'm the first. This is a neat YouTube video of a virus assembling inside a cell, both virus and cell in quotes. Maybe the first thing I fully understand about viruses. I'm going to have to go back to your 101 episodes to get the necessary background for the finer points of your discussions, I know. I've been listening for a couple of months now to all three podcasts and really appreciate those parts that I can understand. Thanks so much for doing them. Peter is from Sydney, Australia. So this is a video. Go ahead.
I'm sorry. Go ahead. No, go ahead.
It's a video of a guy with a big cage rolling around what look like subunits of viruses. They're about 8 inches in diameter. And I guess they have magnets on them, and eventually they all come together to form a virus particle. Right?
Right, and then he does a version of the same thing in a little jar,
Yeah.
miniaturized version.
Cool.
So, I mean, I think assembly in a cell is probably more efficient, but because it took him a long time to do this, and and there's a fade in the middle of the video where they probably cut out.
Right.
The video guy was saying, "Is this ever going to finish? Come on!" So, but it illustrates the idea that the subunits of a particle have all the information in them to assemble and form a virus particle, and that, of course, is the basis for virus-like particle vaccines, like the human papillomavirus vaccine and the Hep B vaccine, etc. So that's pretty cool. Thank you for that, Peter.
That previous pick, Evolution and the Natural History of Animal Skeletons, stripped down, it hypes this book, right? That is evolution as in skeletons.
Yes.
And this was in the queue last week or something like that.
Yes.
I bought the book.
Oh, yeah?
It just looked too cool. So now it's sitting on my coffee table. I don't know if I'll ever read it, but the pictures are awesome.
All right.
Okay.
Cool.
It's got a bunch of skeletons in action and a whole bunch of them.
Well,
and
maybe
you can see the relationship.
maybe you'll make it a pick. The book.
What, the book?
Yeah,
Oh, okay.
If you
It's
like
sort
it.
of a pick within a pick. Okay.
it's okay. It's what's the word? It's
Oh man, I just deleted it. It's back here.
turducken.
Yeah.
A turducken.
A book within a book. Okay, that'll do it for TWiV 179. You can find us at iTunes and the Zune Marketplace, and also at twiv.tv. We do have an Android app which you can use to stream TWiV to your cell phone, either iPhone or an Android device. You can find that over at microbeworld.org. And we do love your questions and comments. We're going to have to do another all-email episode soon, but send them to twiv@twiv.tv. Trudy Radu is at the CDC. Thanks for joining us, Trudy.
Thank you.
And I understand that you have a little project you want to tell us about.
Yeah. So one night I was driving home and I was listening to TWiP, which is This Week in Parasitism. And as we all know, there is no better storyteller than Dixon Despommier. So, yeah, he's very interesting. So while he was telling some story about hookworm, I think, and then went off on a tangent about some person who was involved in some discovery, as he often does, it occurred to me that there really isn't a podcast out there about scientific history. So as soon as I got home, I checked the internet. I emailed you, Vincent. I asked you if you knew of any, and sure enough, there doesn't seem to be any. So what's the motto? If there isn't one, then you should start one, right?
Sure. That's right.
Yeah. So I immediately got to work. Yeah.
Go ahead.
I immediately got to work on it, and I already do a lot of reading about this topic in my spare time anyway. So now, instead of just reading for leisure, I'm actually taking notes while I'm reading. So scientific history is by far my favorite genre. And at first I thought about doing the podcast by myself, but out of all the podcasts out there, I really prefer the ones that consist of a conversation, except for Mark Crislip. He's funny. But so I recruited my husband. He's not a scientist, but he's very interested in science, and he's very inquisitive, and he's a local comedian. So he will definitely inject his humor. Plus, he knows all the technical stuff, so that helps.
Great.
So we decided to call it Sequencing Science. We have a website, sequencingscience.com, and we're on both Twitter and Facebook. The show will be about 30 to 45 minutes tops bi-weekly. And listeners can send in questions and comments, just like on any other podcast. And it's really meant to be an edutainment tool, not just for the listener, but also for us. So I'm looking forward to learning a lot.
What's the Twitter for that? Sequencing Science?
It's Sequencing Sci. It's SCI. We tried Sequencing Science first, but then we had problems with that, and Twitter wasn't very helpful. So,
So have you released an episode yet?
Yes, not yet. I'm sorry. We have one episode, and we're going to release it this weekend. It's about Darwin.
And you'll be on iTunes as well, I presume?
Yes.
Wonderful.
Cool.
Well, we'll put a link to that in the show notes. And
Terrific.
Go check it out.
I just followed you on Twitter.
Wow, cool.
Thank
We'll
you.
have to do that as well. Sequencing Science. Check
Yes.
it out. Well, thanks for joining us today. It was great.
Thank you for having me.
Pleasure. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Quite welcome. Always a good time.
You bet. Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws and on Twitter, PROFVRR.
You've been listening to This Week in Virology. Thank you for joining us. We'll be back next week. Another TWiV is viral.